Welcome. Today we're diving into the fascinating world of gene expression regulation. Genes are the blueprints for all the proteins in our bodies, but not all genes are active at all times. The process of turning genes on or off or adjusting their activity levels is called gene regulation. This control is crucial for development, health, and adaptation to our environment. In this video, we'll explore the mechanisms that cells use to regulate gene expression, why it matters, and how it impacts everything from growth to disease. Let's start by understanding what gene expression means and why its regulation is so important. Gene expression is the process by which information from a gene is used to synthesize functional products like proteins. It starts with transcription, where DNA is copied into RNA, and continues with translation, where RNA is used to build proteins. Not every gene is expressed in every cell or at every moment. Cells carefully control which genes are active, ensuring the right proteins are made at the right time. This selective expression is fundamental for cell specialization and function. Next, let's look at why regulating gene expression is so vital for life. Regulating gene expression allows organisms to respond to changes in their environment, develop properly, and maintain health. For example, when you exercise, your muscle cells turn on genes that help build more muscle proteins. During development, gene regulation ensures that different cell types form, like skin, nerves, or blood. If gene regulation goes wrong, it can lead to diseases such as cancer or genetic disorders. So precise control over gene activity is essential for life. Now, let's explore the main stages where gene regulation can occur. Gene expression can be regulated at several stages: before transcription, during transcription, after transcription, during translation, and after translation. Each stage offers unique control points. For example, cells can prevent a gene from being transcribed, or they can destroy RNA before it's used to make protein. This multi-layered regulation ensures flexibility and precision. In the next scenes, we'll break down each stage and see how cells use different strategies to control gene activity. Before a gene can be transcribed, its DNA must be accessible. In eukaryotic cells, DNA is wrapped around proteins called histones, forming chromatin. If the chromatin is tightly packed, genes are hidden and can't be read. When chromatin is loosened, genes become accessible for transcription. Cells use chemical modifications like adding acetyl groups to histones to control how tightly DNA is packed. This is one of the first steps in gene regulation, setting the stage for whether a gene can be turned on or off. Epigenetics refers to changes in gene activity that don't alter the DNA sequence itself. Chemical tags like methyl groups can be added to DNA or histones, affecting gene expression. These modifications can turn genes on or off and can even be passed to future generations. Epigenetic regulation is influenced by environment, lifestyle, and development. It's a powerful way cells control gene activity without changing the genetic code. Next, we'll see how transcription factors play a role in gene regulation. Transcription factors are proteins that bind to specific DNA sequences near genes. They act as switches, turning genes on or off by helping or blocking the machinery that reads DNA. Some transcription factors respond to signals from outside the cell, like hormones or nutrients, allowing cells to adjust gene expression in real time. This precise control ensures that genes are expressed only when needed. Let's look at how promoters and enhancers further fine-tune gene regulation. Promoters are DNA regions located just before a gene. They serve as landing pads for the transcription machinery. Enhancers are distant DNA elements that can boost a gene's activity even from far away. Both promoters and enhancers work together, interacting with transcription factors to control when and how much a gene is expressed. This complex network allows for precise and dynamic regulation of gene activity. Next, let's see what happens after transcription begins. Transcriptional regulation is the control of gene expression at the stage where DNA is copied into RNA. Cells use a variety of proteins and signals to start, stop, or adjust the rate of transcription. This is often the most important control point for gene expression. By regulating transcription, cells can quickly respond to changes and ensure only necessary genes are active. Now, let's move on to what happens after RNA is made. After transcription, the newly made RNA, called pre-mRNA, undergoes several modifications before it can be used to make protein. These include adding a cap and tail for stability, and removing non-coding regions called introns. This processing is another key point where gene expression can be regulated. By controlling how RNA is processed, cells can influence which proteins are ultimately produced. Next, we'll see how RNA can be further regulated before translation. Alternative splicing is a process where the same gene can produce different RNA molecules by including or skipping certain sections. This means one gene can lead to multiple proteins, increasing diversity without adding new genes. Cells use alternative splicing to adapt to different needs or conditions. It's a powerful way to expand the genetic toolkit. Let's see how RNA stability also plays a role in gene regulation. Not all RNA molecules last the same amount of time in the cell. Some are quickly destroyed while others are stable and stick around. By controlling how long RNA survives, cells can fine-tune how much protein is made. Enzymes can target specific RNAs for degradation, reducing their levels. This is another layer of gene regulation, ensuring that proteins are produced only when needed. Next, let's look at how translation is regulated. Translational regulation controls how efficiently RNA is used to make proteins. Cells can block translation or enhance it, depending on their needs. Factors like nutrient availability, stress, or signals from other cells can influence translation. This allows cells to rapidly adjust protein production without changing the underlying RNA. Now, let's explore how proteins themselves can be regulated after they're made. After proteins are made, they often undergo chemical modifications such as phosphorylation or methylation. These changes can alter a protein's activity, location, or stability. By modifying proteins after translation, cells add another layer of control over gene expression. This ensures that proteins work properly and are available when needed. Next, let's see how feedback mechanisms help maintain balance in gene regulation. Feedback loops are systems where the output of a process influences its own activity. In gene regulation, feedback can help maintain stable levels of proteins or quickly adjust to changes. For example, if too much of a protein is made, it might signal the cell to reduce gene expression. These loops are essential for homeostasis and adaptability. Now, let's look at how gene regulation varies between different cell types. Every cell in your body contains the same DNA, but different cell types express different sets of genes. This selective gene expression is what makes a nerve cell different from a muscle cell. Specialized transcription factors and regulatory elements ensure that only the right genes are active in each cell type. This allows for the incredible diversity of cells and functions in multicellular organisms. Next, let's see how external signals influence gene regulation. Gene expression isn't just controlled from within the cell. External factors like temperature, light, nutrients, and toxins can all influence which genes are turned on or off. For example, plants may activate certain genes in response to sunlight, while bacteria can switch genes on or off depending on available food sources. This adaptability helps organisms survive in changing environments. Now, let's look at how hormones play a role in gene regulation. Hormones are chemical messengers that travel through the body and influence gene expression in target cells. For example, insulin helps control genes involved in sugar metabolism, while growth hormone affects genes related to growth and development. Hormones bind to receptors, triggering signaling pathways that ultimately lead to changes in gene activity. This system allows the body to coordinate complex processes across many cells. Next, let's see how gene regulation is involved in development. During development, gene regulation guides the transformation from a single fertilized egg to a complex organism. Specific genes are turned on or off at precise times and locations, directing cells to divide, specialize, and form tissues and organs. Mistakes in this process can lead to developmental disorders. The orchestration of gene activity is like a symphony, with each gene playing its part at the right moment. Next, let's look at gene regulation in response to stress. When cells experience stress such as heat, toxins, or infection, they rapidly adjust gene expression to cope. Stress-responsive genes may help repair damage, protect against harm, or trigger cell death if needed. This quick response is vital for survival. Cells use signaling pathways to sense stress and activate the right genes. Now, let's see how gene regulation is involved in the immune response. The immune system relies on precise gene regulation to defend against pathogens. When an infection is detected, immune cells activate genes that help fight off invaders. Some genes produce antibodies while others signal for help or trigger inflammation. Once the threat is gone, gene expression returns to normal. This dynamic control ensures a balanced immune response. Next, let's look at gene regulation in cancer. Cancer often arises when gene regulation goes awry. Genes that control cell growth, division, or death may become overactive or silenced, leading to uncontrolled cell proliferation. Mutations in regulatory genes can disrupt normal checks and balances. Understanding how gene regulation fails in cancer helps scientists develop targeted therapies. Next, let's see how gene regulation is studied in the lab. Researchers use a variety of techniques to study gene regulation, such as DNA sequencing, RNA analysis, and protein assays. These tools help scientists identify which genes are active, how they're controlled, and what happens when regulation is disrupted. Advances in technology have made it possible to study gene expression in single cells, revealing new layers of complexity. Next, let's look at how gene regulation is used in biotechnology. Biotechnology harnesses gene regulation to produce medicines, improve crops, and develop new therapies. By controlling gene expression, scientists can make cells produce useful proteins like insulin or antibodies. Gene editing tools such as CRISPR allow precise changes to regulatory elements, opening up new possibilities for treating diseases. Next, let's see how gene regulation is involved in genetic engineering. Genetic engineering involves modifying an organism's DNA to achieve desired traits. By altering regulatory sequences, scientists can increase or decrease the expression of specific genes. This is used in agriculture to create crops with better yields or resistance to pests. In medicine, it can help produce therapeutic proteins or correct genetic defects. Next, let's look at gene therapy and its reliance on gene regulation. Gene therapy aims to treat diseases by introducing, removing, or altering genes within a patient's cells. Success depends on precise regulation, ensuring the therapeutic gene is expressed at the right level in the right cells and at the right time. Advances in gene regulation are making gene therapy safer and more effective. Next, let's see how gene regulation is involved in stem cell research. Stem cells have the unique ability to become any cell type in the body. This versatility is controlled by gene regulation. By turning on or off specific genes, stem cells can differentiate into muscle, nerve, or blood cells. Understanding these regulatory networks is key for regenerative medicine and tissue engineering. Next, let's look at the role of non-coding RNAs in gene regulation. Not all RNA molecules code for proteins. Non-coding RNAs, such as microRNAs and long non-coding RNAs, play important roles in gene regulation. They can block translation, degrade messenger RNA, or influence chromatin structure. These molecules add another layer of complexity to gene expression control. Next, let's see how gene regulation can be disrupted by mutations. Mutations in regulatory regions of DNA can have major effects on gene expression. A single change in a promoter or enhancer can turn a gene on or off at the wrong time or in the wrong place. Such mutations can lead to diseases, developmental problems, or even evolutionary changes. Understanding these effects helps researchers identify the causes of genetic disorders. Next, let's look at gene regulation in evolution. Changes in gene regulation are a major driver of evolution. By altering when and where genes are expressed, organisms can develop new traits or adapt to new environments. Many differences between species are due to changes in regulatory elements rather than gene sequences. Studying these changes helps us understand how life evolves and diversifies. Next, let's look at gene regulation in plants. Plants rely on gene regulation to respond to light, water, temperature, and pathogens. For example, genes controlling flowering are turned on by changes in day length. Plants also use gene regulation to defend against pests and adapt to stress. Understanding plant gene regulation helps improve crop yields and resilience. Next, let's see how gene regulation works in bacteria. Bacteria use simple but effective systems to regulate gene expression. The lac operon is a classic example, allowing bacteria to turn on genes for lactose digestion only when lactose is present. These regulatory systems help bacteria adapt quickly to changing environments. Studying bacterial gene regulation has provided key insights into basic genetic principles. Next, let's look at gene regulation in viruses. Viruses rely on host cells to reproduce, but they also regulate their own genes. Some viruses can remain dormant for years, turning their genes on only when conditions are right. Others rapidly express genes to hijack the host machinery. Understanding viral gene regulation is important for developing antiviral therapies. Next, let's look at gene regulation in response to nutrition. Nutrients can directly influence gene expression. For example, the presence or absence of certain vitamins or minerals can turn genes on or off. This allows cells to adjust metabolism and growth based on available resources. Nutritional gene regulation is important for health and disease prevention. Next, let's see how gene regulation is involved in circadian rhythms. Circadian rhythms are daily cycles in physiology and behavior, controlled by gene regulation. Certain genes are turned on or off in a 24-hour pattern, helping coordinate sleep, metabolism, and other processes. Disruptions in circadian gene regulation can affect health and well-being. Understanding these rhythms helps explain how our bodies keep time. Next, let's look at gene regulation in age. As we age, gene regulation changes. Some genes become less active while others may be overexpressed. These shifts can contribute to aging and age-related diseases. Researchers are studying how to maintain healthy gene regulation throughout life with the hope of promoting healthy aging. Next, let's look at gene regulation in brain function. Brain relies on precise gene regulation for learning, memory, and behavior. Neurons turn on or off specific genes in response to activity, experience, or injury. Disruptions in gene regulation can lead to neurological disorders. Understanding these processes is key for developing treatments for brain diseases. Next, let's look at gene regulation in metabolism. Metabolic pathways are tightly controlled by gene regulation. Genes involved in breaking down food, storing energy, or building molecules are turned on or off as needed. This ensures efficient use of resources and maintains balance in the body. Disruptions in metabolic gene regulation can lead to diseases like diabetes or obesity. Next, let's look at gene regulation in reproduction. Gene regulation is essential for reproduction. It controls the development of eggs and sperm, guides fertilization, and supports early embryonic growth. Hormones and signaling pathways coordinate these processes by turning genes on or off at the right times. Understanding gene regulation in reproduction can help address fertility issues and improve reproductive health. Next, let's look at gene regulation in disease resistance. Gene regulation helps organisms resist diseases by activating defense genes when needed. In plants, for example, certain genes are turned on in response to pathogens, producing proteins that fight infection. In animals, immune genes are regulated to respond to viruses or bacteria. This dynamic control is key for survival. Next, let's look at gene regulation in synthetic biology. Synthetic biology uses engineering principles to design new biological systems. By building custom regulatory circuits, scientists can program cells to perform specific tasks like producing drugs or cleaning up pollution. Precise gene regulation is essential for these synthetic systems to work reliably. Next, let's look at ethical considerations in gene regulation research. Research on gene regulation raises important ethical questions. How should we use our ability to control genes? What are the risks and benefits of gene editing or therapy? Ensuring responsible use of these technologies is crucial for society. Scientists, policymakers, and the public must work together to set guidelines and address concerns. Next, let's look at future directions in gene regulation research. The field of gene regulation is rapidly advancing. New technologies like single-cell analysis and CRISPR are revealing deeper insights into how genes are controlled. Researchers are exploring ways to fine-tune gene expression for personalized medicine, agriculture, and environmental solutions. The future holds exciting possibilities for improving health and understanding life at its most fundamental level. Next, let's summarize what we've learned. We've explored the many layers and mechanisms of gene expression regulation, from chromatin structure to feedback loops and from development to disease. Gene regulation is essential for life, enabling cells to adapt, specialize, and function properly. Advances in this field are opening new doors in medicine, agriculture, and biotechnology. Understanding gene regulation helps us appreciate the complexity and beauty of biology. Let's wrap up with some final thought. Gene expression regulation is a cornerstone of biology, affecting every aspect of life. From health and development to evolution and technology, understanding how genes are controlled is key to unlocking new discoveries and solutions. As research continues, we'll gain even greater insight into the intricate dance of genes and the potential to shape our future. Thank you for joining this journey into the world of gene regulation.